Good morning, everyone. Welcome to the latest webinar in Teagasc's Research Insight series. This series focuses on addressing the challenges and opportunities in the agri-food sector and showcasing the latest in Teagasc research. Today's webinar is brought to you from the Teagasc Food Research Programme, which is based at Teagasc Moore Park and Ashtown. Today we'll focus on food structure, digestion, and the gut microbiome. We have three speakers, Dr. Laura Mascaraque, Dr. André Brodkorb, and Dr. Ciara O'Donovan. And over the course of these three presentations, we will travel inside a powder particle using virtual reality. We're going to see inside the stomach of one of our researchers, and we're going to learn about the bacterial cells that outnumber us in our own bodies. Following the three presentations, we will have a question and answer session. If you have any questions for our presenters, please type them into the Q&A box and include the name of the person the question is addressing. To Dr. André Brodkorb. André is a Principal Research Officer at Teagasc Moore Park. His research interests are in vivo and in vitro gastrointestinal models, the relationship between food structure and function, and he is also a member of the Infogest Cost Action, which is a European-wide initiative which researches food digestion. So over to you, André.
Excellent. Thank you for this kind introduction, Maureen. So as Maureen said, my research interest or my role here in the Teagasc Food Research Center in Moore Park is to follow the structure and function of food from the source, and that might be on the land or it might be with animals or marine sources, through to processing, and this may be cooking or industrial processing, to storage on the shelf life, and then consumption of food and the digestion, the gastrointestinal digestion. So this is the topic of today's talk, and I would like to tell you a little bit about gastrointestinal digestion, and I would take one example. What happens when we drink this glass of milk? Okay, so as soon as food touches your lip, and that might be a solid food or liquid food, a whole chain reaction of chemical, physical, and biochemical reactions will take place. If it's solid food, you have to masticate, you have to chew the food. If it is liquid food, it goes straight through your mouth. However, saliva is still excreted. In saliva, there are a couple of compounds. One of them is amylase, and amylase is an enzyme. And these enzymes are like molecular machines that break down food. In the case of amylase, they break down starch. So once you swallow the food, it will go through your esophagus into the stomach. And in order to help or show what is happening, I have a little helper here. So that is the model torso. So the food will go right through the esophagus in behind the liver into the stomach. I'm taking out the stomach here. So the stomach at the time of birth is about the size of a cherry. And after about 4 weeks, it expands to a size of a walnut. And then it grows throughout your adolescence into a full-size stomach. And that's about it. It might be a little bit bigger. But of course, when you eat food, the stomach can expand greatly to several liters. So once the food passes through your stomach, through the pyloric valve into the small intestine, and this is small intestine, several meters long. At the top of the small intestine is the duodenum, and the jejunum. At the very end is the ileum, and that's really where most of the digestion happens. So especially in the duodenum, and again in the stomach and in the intestine, there are these molecular machines. These enzymes are excreted to break down the protein, break down the fat, and break down the carbohydrates. So when you look at the stomach for us is actually quite interesting, because the journey of the food through the gastrointestinal tract is not linear. So the food actually spends a considerable amount of time in the stomach. That's what it looks like inside. So the food arrives at the top of the stomach, and there it is basically needed. If you have solid food, gastric acid is excreted, and in the case of the stomach, pepsin, which is a protease that breaks down proteins, and lipase, which breaks down lipids or fats. They're excreted here. And at the very end, you have the pyloric valve. So if the food is small enough, so it has to be smaller, the pieces have to be smaller than 2 millimeters. And when the food is digested enough and is acidified enough, it will exit into the small intestine. And this valve is the pyloric valve. And this is actually a very crucial part of the digestion. And as it happened, I have live coverage of a stomach. So a number of years ago, I was working with my colleagues in the Mercy Hospital, and there I had the chance to swallow a very small camera, the size of jelly bean, really. And this camera sends pictures. Sends pictures every 3 seconds. 3 images per second. So here you can see the walls of the stomach. It's actually my stomach. I swallowed the pill. And here at the end, that's the pyloric valve. So that is really the gateway to the small intestine. And later on, I will show you how we use this technology, because this technology is called wireless endoscopy. It is a diagnostic tools used by the medics, and we were the first group to use that technology to try to understand the digestion of food. Now, if you want to understand the digestion of food, what we usually do is we perform human intervention trials. And that means food is given to a number of participants, and then you basically take 3 sets of samples. That is blood, that would be urine, and stool. And these are really the main indicators of how food is digested. However, as you can see, we have no idea what actually happens within the gastrointestinal tract. So in order to understand this, we have to take samples out of the stomach. Now, we have done that again with our colleagues in the Mercy Hospital a number of years ago, and where you put a nasal gastric tube, so a tube through the nose into the stomach, and you can take samples out of it. Now, you may appreciate that this is quite invasive. It's not nice, and some of these human interventions studies, they're quite laborious, they're very expensive, and sometimes you can't do it due to ethical reasons. So for this reason, food research, especially food research, have come up with in vitro laboratory-based digestion methods where we simulate the digestion of food. And here in Moorpark, we have established one of those methods, the Infogest method, where we basically simulate the digestion. First, the oral phase in one tube, then we change the condition to a stomach condition, and then the intestinal conditions. And here on the right-hand side, you see my colleague Daniela Freitas taking out one of those stomach or intestinal samples, and then we can analyze that. So this is a relatively simple method. We published that with a consortium, international consortium last year in Nature Protocol, which is a high impact journal, which is a pretty big deal for us researchers. Now, if you want more sophisticated methods, this is a semi-dynamic method. So where you have you see a vessel right here, a temperature-controlled vessel, and on the left-hand side, you see a syringe, and this syringe adds very small amounts of this enzyme. In this case, it's pepsin, and on the back, we connect this to a titrator, and that adds, the titrator adds the stomach acid, and that way we can nicely and accurately simulate digestion, in this case, of the glass of milk that I showed you earlier. Now, how do we analyze the digestion of the food? Now, here we have one tool which is very commonly used in our laboratory, and that is a technique called gel electrophoresis. Now, you may have come across this in crime programs or CSI, where they use gel electrophoresis to analyze the DNA, so the DNA fingerprint to identify a victim. In our case, we use exactly the same thing, but for proteins. So in this case, there's a gel electrophoresis lane, and each of these bands correspond to a protein. So this particular band is a whey protein, and these bands are caseins. So we basically follow the digestion of the proteins in the stomach as a function of time. And you can see some proteins, like the caseins, they start to be digested, so they're broken down by the pepsin, whereas others seem to be fairly stable, such as whey proteins. Now, the question is, and is a very common question, does the digestion only depend on the macronutrients? So macronutrients are the proteins, the fats, and the carbohydrates. So when you see the example of a glass of milk versus a yogurt, they actually have the same macronutrients, identical proteins, identical fats, and very similar carbohydrates, slight differences, and a pH. So one product is fermented, so has a lower pH, whereas that is pretty much the same, has a ambient neutral pH. So the question is, does it make a difference? And the answer is actually yes, it does make a difference, not just the macronutrients. Another example is whey proteins, and you may have come across this 20 years ago. It was a niche product. Now you see it everywhere. So whey proteins are commonly used in sports nutrition. So what are whey proteins? So whey is a byproduct of the cheese production. So here on the right hand side, you see a cheese plant. This is the cheese curd, which turns into cheese. And this liquid phase here, that is whey. And it's identical if you make it at home. Some people make it at home. This is the cheese curd, and the liquid phase is the whey. And in the whey, you have mainly water, you have lactose, you have a couple of minerals, and you have whey proteins. And in order to get the whey proteins out, you treat this whey with a very fine filter, a membrane filtration, where you filter out the proteins so that you retain the pure whey proteins. Now, why are they so important? Now, we were lucky enough to have a volunteer here in the Teagasc Food Research Center in Moore Park, this Ciara O'Donovan, an all-Ireland hurler that you may be familiar with. And he donated his body to our cause. And poor Shane here on the right-hand side, he swallowed one of those cameras. So on the top left, you see Shane's healthy stomach is empty. And we gave Shane 500 milliliters of commercial whey protein. So at the bottom left, you see Shane's stomach, which is pretty much empty after 90 minutes. So that means all the whey proteins, they are gone out of the stomach. So they're emptied into the small intestine, and there they are rapidly digested. On the top right, again, you see Shane's empty stomach, and we gave Shane 500 milliliters of whole milk. And there the milk arrives, milk is white, you can't see anything. But after a while, it's digested. And to our surprise, after about more than 2.5 hours, we can still see particles of casein. In this case, it's casein in Shane's stomach. So if you compare the right-hand side with the left-hand side, you can easily see the difference between fast proteins, so that's whey proteins, and slow proteins, and that's casein. So you may ask, what's the big deal? Does it matter, fast proteins and slow proteins? The short answer is, for you and me, probably not. However, if you run a marathon, or if you did a triathlon or some vigorous exercise, fast recovery is quite important. So if you want a fast recovery and delivery of proteins, whey proteins are definitely better or faster than the other proteins. If you look at the bigger picture, and that is the digestibility of the proteins, it's a very different outcome. So at the end of the small intestine, about 95% of the proteins are digested. That means they are digested and they are absorbed by the body. So it's relatively high, and most of the animal proteins, so meat and dairy products, have a very, very high digestibility. That is different to plant proteins. Plant proteins, depending on the way they're processed, they might have a digestibility of about 50 to 80%. So it's quite a substantial difference. So last but not least, you may ask, why are you doing this? Okay, so what? And that's a good question. So what? So the answer is, first of all, we have to understand, how food is digested in the body, and we try to simulate in the laboratory, because in the laboratory you can actually understand each individual step, whereas in vivo it is very hard and invasive. So where do we apply this? So one example is a recent example is we were approached by an Irish company. They gave us an ingredient, and they want to know whether this ingredient is easier to digest, or does it digest faster and slower. And we showed it nicely with a relatively simple experiment, set of experiments. We published the work, and this ingredient is now for sale in China as part of a nutritional beverage. The second example that I want to give you is that of probiotics. So a number of years ago, we developed a method to protect probiotics in tiny little capsules, encapsulated probiotics. And in these small capsules, the probiotics, the probiotic bacteria, they survive the stomach. And we were able to demonstrate that very clearly with in vitro and in vivo models. And subsequently, that invention was commercialized by one of the co-inventors, Sinead Blyle. She found a startup company, Anabiotechnology, and it is now based in Carricktool and employs more than 30 people. So that's the real success story. It's an example how we use the digestion models. So that's all I have to say for now, and I'm looking forward to questions later on. And as Moran said, you can type them into the question and answers, and we deal with it at the end of it.
That's great, André. Thank you for that. And again, as André said, if you have any questions for him, type them into the Q&A box, and we'll have a question and answer session at the end of the 3 presentations. Laura is a research officer in Moore Park. Her research interests are food microstructure and microscopy at the National Food Imaging Centre based at Teagasc, Moore Park. Laura.
Thank you very much. As Maureen said, I'm Laura Gomez Mascaraque, and today I would like to introduce you to the research we do in my group in Teagasc, Moore Park, which is mainly focused on studying food microstructure. So today I will start by explaining, first of all, what is food structure? Why are we interested in studying it? How do we do this? And then I will go through a couple of practical applications of what our work can show us. So first of all, what is food structure? Well, we all know that food consists of different components such as proteins, sugars, fats, fibers. So the way all these elements are organized within the food matrix and interact with each other is what we call food structure. And in the same way that our houses will have a different resistance to mechanical stresses when we build them with straw, with wood, or with bricks, the way we design the architecture of our food products will determine how they behave when they are subjected to mechanical stresses, for example, during processing in the industry plant, or even during consumption when we eat them and we chew them. And that's how we feel the texture of the food products. So why are we interested in studying food structure? Well, I'm going to start by telling you about a very interesting experiment that some researchers did. And it consisted on giving some food products, mainly fruits and vegetables, to blindfolded participants. And they were asked to eat them and try to identify them. And it was very interesting because the participants could only identify less than half of the food products when they had been previously pureed to destroy their structure. So this demonstrates that the composition of food products is not enough to define them. We also need their microstructure. Another example is lycopene. Lycopene is an antioxidant present, for example, in tomatoes, and it has many attributed health benefits. And we know that lycopene is better absorbed in our gastrointestinal system when we consume it from processed tomatoes rather than from the fresh fruit. And this is because when we break the original structure of the plant cells, we release the lycopene and we make it more bioavailable. So on the other hand, when we break the original structure of some fruits and vegetables, we can also obtain oxidizing reactions, and this demonstrates that not only the food structure has an impact on the texture of foods and their nutritional value, but also their stability and many other properties of the food product. So that's why food structure is so important, and we are interested in studying it. So now, how do we do it? Well, science is based on observation, so the most direct way of analyzing food structure is by direct visualization. However, only the macro structure of food products can be observed with the naked eye. For example, in this piece of cheese, we can observe some air bubbles, and then what looks like a continuous matrix of cheese where we can't really distinguish the fat from the protein. However, at a microstructural level, we can see that there are different phases with tiny sizes that we can only see with the aid of a microscope, because our eyes, the human eyes, can only resolve objects that have a size of at least 100 microns. And we see objects because there's light that interacts with them. So it is natural to think that microscopy will involve the interaction of light with our samples. And that is the basic concept of the simplest microscopy technique, the light microscopy, in which we just illuminate the sample and collect the light that is transmitted or reflected by the sample. And then we use a set of lenses to magnify the image so that we can see it. And this very simple microscopy technique already gives us a lot of information about food structures. For instance, in this image of starch granules, we can already get information about their morphology, such as the granule size and also the shape. However, sometimes the contrast between the different elements that we want to observe is not enough to use a simple technique such as conventional light microscopy. And in those cases, what we can do is to play a little bit with light and use little tricks to enhance that contrast. One of those techniques is polarized light microscopy. So while normal light vibrates in all directions, polarized light vibrates in only one single plane. And that creates contrast between birefringent materials, basically crystalline materials, and their background. And that's why in this image, in which we also have starch granules, but in this case, we see them shining because starch granules are semi-crystalline. And then we can see that as the temperature increases, this starch is gelatinizing and the granules are not shiny anymore. And these structural changes occur every day in our kitchen when we cook food that contains starch at high temperatures. And this has an impact also on the way that starch will be digested in our gastrointestinal system. This is another example. This is skim milk powder. And milk powder, when we store it at high relative humidities, the lactose in it crystallizes. So again, it appears shiny in this image. So polarized light microscopy is a very nice technique to study this type of phenomenon. Another technique to try to increase contrast between phases and elements in a sample is fluorescence microscopy. So in this case, instead of using normal white light, what we use is a monochromatic light, that is light of a single color. And what we collect is the information only from the elements of the sample that exhibit fluorescence when illuminated with that specific color of light. And the advantage of this is that we can use different selective fluorescent dyes to stain different elements. For example, only proteins or only fats. And this is what we did in these examples. These are 4 images of 4 different types of cheeses. And we stained the proteins with one selective dye and the fats with another one. And we can see the proteins in red and the fat in blue. And we can already see how the structure of all the samples, even if all of them are cheese, are completely different. For example, if we look at mozzarella, it has its characteristic elongated features. And this is because of the cooking and stretching process that it is subjected to when it is produced. And it will also have an implication on the way it will behave when molten. So the food microstructure also has an impact on the application that we can give to those food products. It's not the same mozzarella that we will probably use to melt than cream cheese that we use for spreads. And this is another example of how the food structure determines the functionality of some food products. What we have here are two dairy powders, like infant formula. And usually when we dissolve these powders in water, we don't want to get flakes. We want to obtain a very homogeneous dispersion like milk. So here on the left, we can see what we would consider a good powder from that point of view, because we can see how it clearly and nicely dissolves in water, while on the right-hand side, we have what we would call a bad powder, because it doesn't dissolve as nicely. And if we look at the microstructure, we can clearly understand why, because while this one on the left has its fat droplets, which are in red, nicely encapsulated within the powder particle, in the one on the right, we can see a lot of fat on the surface, which hasn't been properly encapsulated. And this will have implications because the fat will repel the water and then it will avoid its proper redispersion. This is another nice example in which we can observe how skim milk gels in the presence of an enzyme. And we can see that at a microstructural level. So what we did here was basically stain the proteins with a selective fluorescent dye. And we can see at the beginning of the video how the proteins move freely at the beginning until at some point the sample starts gelling and the movement is restricted and a network is formed. And this can clearly be correlated with the rheological properties of the sample, which basically tell us how solid the sample is becoming with time. One other example, so when we build food structures, we always have in mind that at some point they will be broken down because the final purpose of the food is to be consumed. So to understand how we perceive the texture of foods, we can subject the foods to mechanical stresses and observe what happens. Here we have two different gels. Both have proteins in red and fat in yellow. And the difference between them is the pH at which they were produced. So we can clearly see how the one that was produced at neutral pH is very elastic when it breaks, while the one produced at acidic pH is more brittle. And I would like to briefly introduce a last microscopy technique, just to say that light is not the only source of illumination we can use in microscopy. We can also use subatomic particles like electrons for this purpose. And this is how an electron microscope works. So there's a theory of the quantum mechanics that says that all matter can behave like waves. So this basically means that particles can behave like light, but with a much more stronger energy, which means that in the end, we will have a much better resolution when we use an electron microscope than when we use any light microscope. For example, here what we have are some probiotic bacteria embedded in a yogurt matrix, and we can clearly see those microscopic organisms in the image. While if we use a light microscopy, like for example, fluorescence microscopy, all we can see are little dots. And finally, I wanted to say that thank you to the new developments in microscopy techniques and also software for image analysis. Nowadays, we are able to study food structure in its 3 dimensions and even get embedded into the food matrices using virtual reality to actually study all the food structure in its 3 dimensions. And all this information that we get about food structure not only helps us as scientists, but also the food industry to develop better food products with improved characteristics. And that's all I wanted to say for now. Again, you're welcome to ask questions in the questions and answer option, and we will go back to that at the end. Thank you.
Thanks, Lara. That was fantastic. Some really cool images and videos there. So, and again, if you have any questions for Lara, pop them into the Q&A box and we'll get to them after the final presentation. So introducing our final speaker, Dr. Akira Odonovan is a postdoctoral researcher in the Food Biosciences Department at Moorpark. Her research interests are the gut microbiome and the effect of diet and other fermented foods on our gut bacteria. So Ciara, I'll let you share your screen, please.
Okay, good morning, everyone, and thank you so much, Maren, for the introduction. So today I want to talk to you about the gut microbiome. So specifically what it is, what factors can influence our gut microbiome, why we are actually interested in our gut microbiome, how we research our gut microbiome, and finally some changes we can make or some things that we can do to change our gut microbiome in potentially positive ways. So firstly, what is the gut microbiome? So when we speak about microbiomes, we are talking about all the different microbes, so bacteria, viruses, fungi that are living on and within the human body. So when we compare the cells and genes of human to microbial cells and genes, we are in fact more microbial than human. We have many different microbiomes on our skin and our oral cavity, but today I'm going to be focusing on the microbiome in our gut. So by numbers, this is the largest microbiome that we have. And this microbiome is subject to many different changes throughout our lifespan. So when we are born, our gut microbiome is quite unstable and subject to many different changes, but around 5 years of age, this stabilizes, but there are still factors which influence the gut microbiome throughout the rest of our lives. So what you can see here by these circles, each of the colors in these circles represents a different microbe. And as you can see from comparing the different life stages or health stage of these individuals, that the microbes that are present are quite different. How do we actually study the microbiome? So traditionally, agar plates would be used to study this kind of environment that you might see here. And this would involve having an agar and growth conditions that was suitable for all of the microbes present. However, such an agar doesn't actually exist, and many of the microbes that are present in our gut microbiome cannot be cultured. And so when we use these techniques, we are really only seeing the tip of the iceberg. Through using DNA sequencing technologies, this allows us to explore those microbes which are unable to be cultured without the need for actually culturing them. So DNA sequencing-based approaches can either be focused on a specific community, such as those bacterial populations present in the gut microbiome, or we can use whole metagenome shotgun sequencing, where we sequence all of the DNA present in a sample, and then we can look at the community composition, so who is there, as well as the genes and pathways of these microbes, so the actual functionality of these of the microbes that are present in the microbiome. Both of these approaches are incredibly useful. However, most current studies of the gut microbiome does use the shotgun sequencing approaches. But why are we actually interested in our gut microbiome? Well, our gut microbiome is involved with a variety of different functions contributing to health, well-being and functioning, including some of those that are depicted here. So the synthesis of vitamins and short-chain fatty acids, contribution to digestion and energy harvest from food, as well as absorption, the development and stimulation of our immune system, protecting against pathogens, and involvement with the gut-brain axis, which I'll speak a little bit more about later on. So how can we actually change our microbiome? Well, there a range of factors that influence our gut microbiome. These can either be positive influences or negative influences, and some factors might influence our gut microbiome very quickly, but other factors might take a long time to induce any changes. So firstly, I'm going to be talking about some uncontrollable factors, and I say they're uncontrollable in that in our day-to-day lives, there's not really much that we can do to influence these factors, and this includes our genetics, our geography, how we are born, our first foods, and aging. There are also a number of factors which are slightly more within our control. So antibiotics and medications have a huge impact on the gut microbiome. Antibiotics are vital in the society we have today and prevent a number of preventable deaths every year. However, their misuse should be avoided. They do have a big impact on the gut microbiome, including the reduction of diversity. Exercise is a factor that has been established as impacting the gut microbiome, with more about that in an upcoming webinar. Healthy aging. So I mentioned that aging is an uncontrollable factor in that we can't really do anything about the fact that we are aging. However, we can, with behavior modifications, we can control for some of the changes to the gut microbiome. Finally, diet. So diet is one of the factors which is most studied in relation to our gut microbiome, and is the factor that I will be focusing mostly for the rest of this presentation. Depending on the dietary factors involved, these can again be positive or negative changes to the gut microbiome. So industrialization has been found to have a huge impact on the human gut microbiome. So factors such as medication that we spoke about, sanitization, like processing of foods, and a variety of factors like this differ from a more traditional exposure to whole foods and other microbial exposures. And what this does to the microbiome is it basically reduces our diversity. So what we have here is each of these bars represents a different country. On the left, we have more traditional countries moving to those industrialized countries on the right. And what was identified here was that those in traditional countries had a higher alpha diversity in the gut microbiome of those individuals in those countries, while those in more industrialized countries had a lower alpha diversity. Diversity is very important in our gut microbiome, and having a high diversity has been continuously linked with not only gut health, but also overall health of an individual. But perhaps there are some changes that we can make to positively influence these somewhat negative changes to the gut microbiome. So firstly, I'm going to talk about probiotics. So you've probably heard about probiotics. They're live microorganisms that are taken usually either in food or maybe capsule format. Lactobacilli and bifidobacterium are commonly used probiotics. And the function of probiotics is to be delivered directly to our gut, as you can see here. So these are our gut epithelial cells, and the microbes in probiotics, the aim of these is to be delivered here to enact their functions. And some of these include barrier protection, so making sure that this barrier in the gut epithelial cells is strong, preventing the attachment of pathogens here, and also the stimulation of the immune system. So these are some immune cells here, so the stimulation of these. Prebiotics, so slightly different from probiotics. Here we're delivering a substrate to the bacteria that are already present in the gut environment. So in this example, prebiotics are delivered to the bacteria present, and these are producing short-chain fatty acids. So depending on what's delivered, different metabolites or stimulations will occur, but in this instance, short-chain fatty acids are benefiting these intestinal epithelial cells, again through barrier functions and actually providing energy. Many different foods, including some of those that are shown here, are examples of prebiotics, such as artichokes. Seaweed contains prebiotics, and even wine has some prebiotic potential. And depending on the actual food, it is a different type of prebiotic stimulating the environment in a different way. So next on to fermented foods. There are fermented foods produced in virtually every society in the world. Some of these might look quite familiar to you, just cheese or salami. Some of them might be new to you, it's just the beverage tapache. You might have seen kombucha for sale in the shops. This is kind of what it looks like when it's actually being produced. The consumption of fermented foods has been reduced in Western countries. However, due to perceived health benefits surrounding fermented foods, this has been on the increase and it's projected to continue to increase. So I mentioned the perceived health benefits. So if you look up any of the number of fermented foods that are available, you'll be met with a whole host of sites claiming their benefits. However, the research into fermented foods is actually in very early days, and some of these claims should be treated with a lot of caution as there isn't much basis to them. So what are the actual health benefits that have been identified associated with some fermented foods? So these include the prevention or protection against type 2 diabetes, obesity, protection against cardiovascular health, depression, osteoporosis, and the improvement of muscle soreness post-training. A lot of these health benefits that have been identified are association studies. So for example, those who eat more fermented foods are less likely to be obese, or looking at individual components of a fermented food instead of the whole fermented food. And so the research is still ongoing. Let's talk about kefir. So you've probably heard about kefir. So kefir, as you can see being made here by my colleague John, is a fermented beverage. It's made by the fermentation of milk with a consortium of bacteria and yeasts. They're in this bacteria, in this cauliflower kind of like clump. And the microbes that are present in kefir change throughout the fermentation depending on how long it's left. And there's very few microbes actually present in kefir. And this makes it very useful in investigations of fermented foods. And it has been studied in a number of studies. So what do we actually know about kefir? So kefir has been identified as being potentially probiotic. So in one investigation of kefir, one of the members of kefir, Lactobacillus kefir was identified as being able to survive gut transit, colonize the gut, and inhibit pathogens. So these factors make at least part of the kefir potentially probiotic. But does this actually hold true? Does this happen when we consume kefir? So in feeding kefir to healthy adults, it was identified that these are 3 species which are commonly found in kefir. The grey that you can see here is before treatment, the red is after treatment, and these species were found to increase after the consumption of kefir. So that identifies kefir as being potentially probiotic, and also that potentially some of these microbes can actually colonize the gut. What about some of the health claims surrounding kefir and other fermented foods? So here, kefir was investigated in terms of its relationship to the gut-brain axis. So mice were fed kefir, 2 different kefirs, and what was found was that one kefir had an antidepressant-like effect, but another kefir increased reward-seeking behavior. The changes in the microbiome were very similar in the species that were increased or decreased between these 2 kefirs. And in relation to the actual gut-brain axis, so there was these behavioral differences and some microbes changing. The microbes that were present were identified as having GABA or a serotonin precursor production potential. And these elements have been identified as being decreased in anxiety or depression. And so it's potentially that some of the differences that we're seeing in the behavior side were at least partially being moderated by the microbes that were present. So to conclude, today I kind of wanted to cover a couple of questions. So firstly, what is the gut microbiome? So it's those microbes existing in our gut. So they're present in huge numbers and they change throughout our life and with different disease factors. Why we're actually interested in it. So it's really important for our overall health and existence, and it also contributes to many functions. How we change it. So a range of factors influence our gut microbiome. Today, I mainly focused on those dietary factors such as probiotics, prebiotics, fermented foods, but as you could see, a lot more research is needed, especially in the arena of fermented foods. I'd like to thank all the members of Vision 1, where I'm based, especially those whose work I presented today. Thanks to the organisers for inviting me to participate, and thank you so much for listening. I'd be happy to take any questions you have for me now. Thank you so much.
Thanks, Ciara. And we'll start our Q&A session now. So we've got loads of questions coming, keep them coming in for the three speakers. I'm going to start off with André, because we've got a few questions on the in vitro model. So first of all, how closely does the in vitro model match in vivo conditions?
Yeah, so there are several different models. Some are pretty simple and some are fairly complex. So the simpler models, obviously, you know, they can simulate a certain amount, give you a certain amount of information. However, it is actually quite good if you have different foods to directly compare them. Okay, if you do it carefully enough, you get a lot of information out there. And the Infogest method that I mentioned earlier, which is a simple method, has been validated for several products, and it is surprisingly accurate. If you're looking at the kinetics of digestion, so meaning as a function of time, you probably have to go to more sophisticated models. And we have these in place as well.
And kind of following on from that then, can you bring a product to market just using these in vitro models, or do you generally need an in vivo study as well to back up your data?
I think it depends on the claim. If you have a big claim, you know, it helps, you know, to cure A, B, and C, there's no way around an in vivo study, a human intervention study. Okay. If you want an indication, you know, how it behaves, I think in vitro models are quite useful, very useful, because you can perform them in the laboratory and you don't have to go to human intervention trials. But it depends on what is the information. If you want to get a product on the market with a strong claim, you have to go in vivo. You have to.
Thanks, André. I'll move on to a couple for Laura now. First of all, we hear a lot about viruses these days. Can you pick up small, even smaller particles by using the microscopy methods you described, such as viruses?
Oh, yeah. For example, electron microscopy has enough resolution to actually analyze viruses. Some of the images that are going around now about the coronavirus with the little spike proteins, those are obtained with electronic microscopy. And we also have other microscopy techniques that can even go to further resolutions. And definitely we can see nanoscopic objects with microscopy.
And another question came in asking, can you use atomic force microscopy to find out the internal structure of zero gel made out of flour?
Well, the key word here is internal, because atomic force microscopy is another technique I didn't talk about in my talk. There are plenty of microscopy techniques, and all of them have different advantages and disadvantages. Atomic force microscopy in particular has a very high resolution, but it is used to study the topography of materials. So you would be able to observe the surface of the serogel, but not the internal structure. So to observe the internal structure, there are other techniques that you can use. Probably cryo-SEM would be a good option, because in that one you can cut in you first freeze the sample so that it doesn't alter its structure, and then cut the internal structure, and then you can analyze that internal structure.
Okay. For Ciara, you talked a lot about effects of different foods and diet on the gut microbiome. Are there any studies on the effect of a vegan diet on the microbiome?
So yeah, I think there is some investigations of a vegan diet. I guess in general, a vegan diet tends to have a lot more fiber, and that individuals are eating much more fruit and vegetables. And there's a lot of investigations of comparing a fibrous diet to a vegan diet. So I think, or comparing a fibrous diet to a non-fibrous diet. So I think when you're comparing a vegan diet, that is obviously a big kind of confounder that you have there, that's probably one of the, so I think you'd have to kind of compare like to like, but there are some investigations that it kind of increases diversity again because of the high fiber kind of aspect.
I might come in there. So if you're looking at the vegan diet, my recommendation would be educate yourself, do some research on with reliable sources. So not necessarily internet, but reliable sources. So there's nothing wrong with vegan diet. If you go that way, just educate yourself. I mentioned earlier the digestibility of proteins. So you probably have to treat some of the food, cook some of the food to make it available. But again, educate yourself because it is not a complete diet. I think vitamin B12 is missing, a few micronutrients, but not many. You know, just be sensible. And
for Ciara as well, in the consumption of fermented foods, are there any kind of detriments to it? As if the question here asks, you know, could there be too many short chain fatty acids produced by the bacteria, or has that ever been investigated?
So I don't know if that is potentially an impact. I think it is important if you're making fermented food yourself that you're very cautious, because if you're fermenting food, you're obviously leaving it, and there is a big potential that that in itself might make the food unsafe to actually consume, and it might make you sick in that kind of aspect. If you're just eating fermented foods, if there's too much that can happen to your gut microbiome, I don't know if that's been thoroughly investigated, but yeah, I think if you are consuming fermented foods, make sure that if you're making them yourself, be really conscious about making sure everything is very well cleaned. Do a lot of research into how you clean the vessels that you ferment in, and making sure, like for instance, if you're using like brine, that all the vegetables or whatever you're fermenting is under the brine, because otherwise you'll have a lot of maybe issues that are kind of negating the potential benefits of actually eating the fermented food.
Okay, so we got a good question here addressed to both André and Ciara. People usually relate the gut microbiome to the large intestine and study that pretty intensely with in vitro models. But we know today that we also have a microbiome in our small intestine. Can we simulate that in models as well?
I can start with that. So for the food digestion, what I mentioned earlier, the microbiome, the bacteria is probably not quite as important. So for the digestion of proteins or for fats and so forth. However, there's a drive to simulate the gut microbiota in the small intestine. It is difficult, it's still difficult, because if you compare the small intestine with the large intestine, it's like a rapid stream leading into a lake. Okay, so it is transient. The small intestine is transient, so it's constantly changed. Where the large intestine, you know, is more stagnant. It stays there for 24 hours or longer. So it's a little bit different. Maybe Ciara, if you want to say something about that.
Yeah, so I guess in theory it is potentially possible to kind of investigate those kind of the interactions there. Like as André kind of mentioned earlier, there's kind of like a lot that we maybe don't know in terms of like the microbiome in the actual small intestine, because it's, when you're doing those kind of investigations, it's not very comfortable for people to be like sampling those kind of areas, whereas like other like environments, it's much easier to sample. So in a lot of investigations, we're focusing on kind of the fecal microbiome when we're talking about the entire gastrointestinal microbiome. So I think that's an important factor to kind of recognize, and I think there's definitely a bit of a maybe gap there that kind of needs to be addressed.
Yeah, still early research, so there's a lot of there's a knowledge gap there, and I'm not aware of any simulation, in vitro simulation. Probably some very simple ones, but not a proper validated.
Yeah, nothing that kind of covers everything, because obviously you have a lot of like immune interactions that are kind of more difficult to simulate, and as André said, like a lot of change and fluctuations that are kind of difficult to control for it properly.
For Laura, in fluorescent microscopy, how do you color the specific components such as the proteins or the fats?
Yeah, so what we do is to add selective dyes, and what I mean by selective is you have some component that is fluorescent, and if, for example, in the case of fats, we select a dye that is lipophilic, which basically means that it has high affinity for fats. So this dye will penetrate the food and go only to the regions rich in fats. While, for example, for the protein, we would use a dye that is water soluble, so it won't penetrate into the fat and will attach to the protein because it will have high affinity for the protein. So then we can map the areas where the different fluorescent dyes are, and we can see where then the protein is and where the fat is.
Sounds good. So I'm just going to finish off on the last question for all of the presenters. We have a 6th year biology class listening in. So thanks for joining us this morning. And they have asked our 3 researchers to just explain to the students, in college, did you focus on biology, chemistry, or how did you get to the area of research you're in there?
Ladies first. Go on, Laura.
Yeah, so, well, at school, I had to select if I wanted to study science. And of course, I knew that I wanted to go for science, and I already liked chemistry a lot. So then when I moved to university, I studied chemical engineering. And during the time that I was studying, I had the opportunity to start doing some practical research with the professors at university, and that was already in the area of food. And then I did my master's in food science and my PhD also in food science.
Very good. So I'm German, so I did my degree in Germany. It was a chemistry degree, really hardcore, chemistry. And the reason I like chemistry, that's why I started. And subsequently, I did a PhD in cancer therapy. So I did my PhD with 5 milligrams of samples. When I started in food science, very new area again. My background was very different. My first experiment with 1 ton of food, so very different. But in our group, we have all kinds of different backgrounds, from biology, microbiology, chemistry, biochemistry. My advice to the 6th-year biology students would be, do what you like to do, really what you like to do. And it will pan out. You might start with something that you think you like, but it turns out to be quite different, and you will find your path. Science is definitely a good career. And you can see from the panel, there's space for the girls, for women in science. We have a lot of female scientists here in Moore Park. So there's a place for you in science.
And Ciara, just maybe 1 or 2 lines from you, because we'll wrap up in a minute.
Yeah, sorry. So yeah, when I was in secondary school, I did biology for my leaving cert, and then in college, I did nutrition and health science. I kind of chose that specifically because there's a lot of kind of biology related modules, and I was just really interested in that. And then I went on to do a PhD in microbiology, mainly looking at the gut microbiome. And so yeah, just to kind of follow on from what André said, like I just kind of always kind of followed routes of things that I was really interested in, and it just kind of makes your days kind of a little bit easier if you're enjoying a little bit what you're interested in, what you're actually studying. So that would be my big tip to all these 6 years.
Thanks, Ciara. And it's Bridgetown College are the class that joined us, so thanks for joining us this morning. So we'll wrap up the Q&A session there. We didn't get to a lot of the questions, so we will get the researchers to answer them after the webinar and put the answers up on the website. In 2 weeks' time, we have another webinar coming from the food program, where we'll continue our story of food, digestion, and the gut microbiome from Dr. Linda Giblin, Dr. Maria Hayes, and Dr. Orla O'Sullivan. So I'd like to thank you again for joining us this morning and for all your questions. I hope you'll join us again in 2 weeks' time. Until then, Gurv Mila Malgut, August Lawn.
Bye.
Thank you very much. Bye.
Bye. Thank you.